Welcome to The Explainer. Today we're diving into a fascinating paradox that's right at the heart of genetics. How is it that having extra chromosomes can create these incredible superplants, but in humans it can lead to serious disease? It's really a tale of two very different genomes, and our story actually starts with a fruit you've probably eaten this week.
So, think about the last banana you ate. It was sweet, easy to eat, and I'm willing to bet it didn't have any of those big, hard seeds you'd find in a wild banana. So why is that? The answer isn't just about clever farming, it's a surprising genetic secret that makes the modern banana even possible.
And here's that secret, a condition called autopolyploidy. You see, we humans are diploid. We have two sets of chromosomes. But the common banana you get at the store is a triploid, meaning it has three. When that plant tries to make seeds, those three sets of chromosomes just can't pair up neatly. Think of it like trying to get everyone a partner for a dance when you have an odd number of people. It's just chaos. For the banana, that genetic chaos means it can't produce viable seeds.
And this brings us to our first big idea. For plants, having these extra sets of chromosomes, what we call polyploidy, it's not a bug, it's a feature. In fact, it's a kind of genetic superpower that has shaped the entire plant kingdom.
And I mean it when I say it's shaped the plant kingdom. This isn't some rare thing. We're talking about 30 to 35% of all flowering plants, and a truly staggering 70 to 80% of all grasses are polyploids. Now, compare that to the animal kingdom, where the condition is almost non-existent. And that huge difference is really the central mystery we're unpacking today.
Okay, so duplicating their own genomes is one thing, but plants can take this a whole step further. They can pull off a kind of genetic fusion, combining entire chromosome sets from totally different species. This process is called allopolyploidy, and it's basically a recipe for building brand new, incredibly powerful hybrid plants.
Here's how it works. So imagine you cross two different species. Let's call them plant A and plant B. Now their offspring is usually sterile because the chromosomes from each parent don't have a matching partner to pair up with for reproduction. But then something absolutely remarkable can happen, a spontaneous doubling of the entire genome. All of a sudden, every chromosome from plant A has a perfect partner, and so does every chromosome from plant B. And just like that, the hybrid plant becomes fertile. In a single evolutionary moment, a new species is born.
A perfect example of this is triticale, which is a hybrid of wheat and rye. See, wheat gives you high protein, but it doesn't do well in poor soil, and it lacks an essential amino acid called lysine. Rye is kind of the opposite. It's super tough and high in lysine, but lower in protein. Well, the triticale hybrid gets the best of both, the high protein from wheat and the toughness and high lysine from rye. A true superplant, created by combining two different genomes.
And here is where our story takes a huge turn. If polyploidy is so amazing for plants, you have to ask, why don't we see this in animals? This is where the rules that build superplants just completely break down when it comes to us.
The difference really couldn't be more dramatic. For plants, extra genomes can mean strength, new species, better crops. For animals, it's a total disaster. It almost always results in sterility, like with mules, or the embryo simply isn't viable at all. You see, animal development depends on this incredibly precise dosage of genes, and doubling everything just throws all those delicate biological pathways into complete chaos.
So, if whole extra sets of chromosomes are a non-starter for animals, what about something smaller? Let's zoom in from entire genomes to the impact of just one single extra, or one missing chromosome. This is a condition called aneuploidy, and for animals, this is where you really see how much our bodies rely on incredible precision.
So what is aneuploidy? Well, it's a change in the number of chromosomes, but on a much, much smaller scale. Instead of a whole extra set, it just means having one extra chromosome or one missing chromosome. And this usually happens because of an error called nondisjunction. Basically, the machinery that's supposed to pull chromosomes apart when making sperm or egg cells messes up.
Now, when that error leads to one extra chromosome, we call it a trisomy. So an individual cells have 3 copies of a certain chromosome instead of the usual 2. The most well-known example of this in humans is trisomy 21, which is the genetic basis for Down syndrome. The flip side of that, having one missing chromosome, is called a monosomy. Now, for our non-sex chromosomes, the autosomes, this condition is almost always lethal, really, really early in pregnancy. It's just a powerful reminder that human life is balanced on this razor-thin genetic edge, where the exact amount of information is absolutely critical.
So where do these errors usually come from? If we look at the data for Down syndrome, it's pretty striking. Roughly 80% of the time, that nondisjunction error, that failure of chromosomes to separate correctly, happens while the mother's egg cell is being formed. And that one piece of data leads us to a really critical question.
And that brings us to our final section, unpacking that well-known but often pretty misunderstood link between a mother's age and how often we see conditions like Down syndrome.
So, why does this correlation even exist? Why does the risk go up as a mother gets older? It's not a simple question, but the answer is buried deep inside the really unique biology of how human egg cells develop. The answer is in this incredible timeline right here. In human females, the process for making egg cells, meiosis, actually starts before they're even born. But then it just stops. It pauses, arresting the cells right in the middle of the process. And they stay in that suspended animation for decades, only starting up again when an egg gets released during ovulation, which could be 16, 30, or even more than 40 years after the whole thing began.
So, that decades-long pause seems to be the key. The leading theory is that the molecular structures holding the chromosome pairs together, you could think of them like aging glue. They can just become a bit more fragile over all those years. So when meiosis finally restarts at ovulation, decades later, those weakened connections are more likely to fail. The chromosomes don't separate properly, and that's the non-disjunction error that can result in an egg cell with an extra chromosome.
So from a seedless banana all the way to the very blueprint of human life, it's clear that the number of chromosomes has a massive impact. It really shows this fundamental split in the strategies of life. For plants, more can be better, it can be a source of strength. But for animals, it is all about precision and a very delicate balance. In the delicate blueprint of life, a single extra or missing chromosome really can change everything.